Hello everyone. So today's video is on DNA replication. So we're going to be looking at, first of all, the DNA double helix as a structure, that kind of helical structure, and then we're going to move on to the mechanisms behind DNA replication, and it's for eukaryotic organisms, not prokaryotic organisms. So I've got here a two-dimensional cross section of DNA to kind of help people grasp the kind of anti-parallel and complementary nature of DNA before we move on to the replication process. Now, DNA itself is anti-parallel. That helical structure contains two DNA polynucleotides, and both, as you know, that phosphate sugar backbone is critical in that kind of polymerization process, that those hydroxyl groups and phosphate groups. Now, it's anti-parallel in nature, which means one of the polynucleotide strands will run in a 5 to 3 prime direction and the other will run in a 3 to 5 prime direction. What do we mean by 5 prime? Well, the numbering just refers to the carbon and the respective functional group that's facing the N-terminal of the polynucleotide. So in this case, we know on the deoxyribose sugar, that fifth carbon is critical in forming that phosphoester linkage with that phosphate group. And conversely, that third carbon on the deoxyribose sugar contains that hydroxyl group, which is really important as acting as a nucleophile during the kind of nucleic acid polymerization process. Now, so you can kind of see that the other strand, the anti-parallel strand, that other complementary strand will be running in the opposite direction. So where you've got at the top on the left-hand side, you've got a 5 prime phosphate sticking out that functional group at the terminal end of the DNA, and then on the other anti-parallel strand, you have that hydroxyl group that's sticking out, that's on the third carbon. And you'll see this, and you can kind of predict the directionality of DNA because of that, because of those functional groups. Now, DNA is also complementary. The pairing is referred to as complementary in nature. And all this means is that the nitrogenous bases that we discussed in the previous video will, a respective pyrimidine will hydrogen bond with its respective purine. And as you know, the pyrimidines are just the six-membered rings, and the purines are the nitrogenous bases that contain both an imidazole ring, so a 5-membered ring, and a 6-membered pyrimidine ring. Now, both purines and pyrimidines, as we know, they're heterocyclic in nature. Now you can see that adenine and thymine will always find each other. That's the complementary pairing. It stays consistent across the DNA double helix, and guanine and cytosine respectively will also hydrogen bond together. Now, hydrogen bonding is a really critical kind of concept in understanding the DNA double helix and the complementary base pairing. So I'll go over hydrogen bonding in the next slide, but just to kind of look, you can see that adenine and thymine are hydrogen bond together. That carbonyl group on the thymine is hydrogen bonding or acting, that oxygen is acting as a hydrogen acceptor, and that amine group on the adenine, those two, one of those hydrogens is likely to be electropositive, so therefore it's going to be acting as our hydrogen acceptor. And you can see that guanine and cytosine contain 3 hydrogen bonds, making them much more thermodynamically stable. The adenine and thymine is much less thermodynamically stable, which is really important in initiation of the replication process. Those adenine thymine rich areas kind of act as that kind of origin of replication site where DNA replication initiates, due to that, a reduced thermodynamic instability. It gives our enzymes, our helicase, our MCM2 enzymes, a head start when breaking that double helix apart, and that breaking of the double helix is critical during DNA replication. So I just thought I'd kind of reinforce this idea of hydrogen bonding to help people understand this kind of complementary pairing between the nitrogenous bases and the role their functional groups play in kind of maintaining and creating this very strong, stable chemical medium that is DNA. And a lot of that is largely attributed to hydrogen bonding. Now, hydrogen bonding is a weak non-covalent bond between a hydrogen atom attached to an electronegative atom, usually on a functional group, okay, and another electronegative atom, okay, on another functional group on a neighbouring molecule. And hydrogen bonding occurs between polar molecules, and DNA is polar, just like water, for example. And all polar means is there's an uneven distribution of electrons across the molecule, okay, usually resulting in lone pairs of electrons, which creates a more electro negative or delta negative side of the molecule and a more electropositive side of the opposite side of the molecule. So there's an uneven distribution of electrons, and this essentially allows for hydrogen bonding. And you can use water as an example. It's a much more simpler molecule than DNA. You look at H2O, for example, you've got one oxygen and you've got those two hydrogen atoms. Now, what's happening is because of the size of oxygen's atomic nuclei, there's going to be more pull. Okay, so it's going to be holding those electrons, giving it lone pairs of electrons that are going to make that kind of oxygen head, if you like, electronegative. It's therefore acting as a hydrogen acceptor for a neighbouring water molecule. Okay, that delta positive, that electropositive hydrogen on that neighbouring molecule has formed a weak intermolecular force between that delta negative oxygen, and that hydrogen that we're talking about is also going to be electropositive because it's also bonded to an oxygen on the neighbouring water molecule, and that oxygen is holding those electrons. Okay, that oxygen becomes electronegative, and that oxygen can also act as a hydrogen acceptor. And you can see how when that happens on a large scale, it creates a very strong, stable chemical medium. Okay, and this essentially gives water can attribute its biological properties to hydrogen bonding. Okay, it's very cohesive, it's very sticky, it's got a high specific heat capacity, and that essentially because of its hydrogen bonding is essential for life. Hydrogen bonding also governs the quaternary structure of proteins. It governs protein folding and shape, which we'll talk about when we talk about protein synthesis, amino acids, and it's a very important concept in biochemistry, and it's no different to the kind of complementary pairing you see that happens between the nitrogenous bases in the DNA. So you can look at, say, for example, the carbonyl group you've got in thymine, that C double bond O. Yes, carbon is more electronegative than oxygen, but oxygen has less shielding, remember, so there's more exposure to that. It's got more exposure to that kind of atomic nucleus, if you like, so it's going to be more pull felt. Okay, so it's going to be holding those electrons from that carbon, if you like, and it's going to become more delta negative, and it's going to be capable of acting as a hydrogen acceptor. Okay, and that amine group that's on the adenine, for example, that amine group, that NH2 group, you see nitrogen's a lot more electronegative than hydrogen. It's going to be holding those electrons, making that hydrogen more electropositive. Okay, it's more capable of being a hydrogen donor. So you're going to get a weak intermolecular force between the hydrogen on the amine group of one purine base, and then the delta negative oxygen or electronegative oxygen on that carbonyl group on the adjacent or opposite pyrimidine. And that allows for that kind of that governs that kind of complementary pairing, is that hydrogen bonding. So I've chosen another two-dimensional cross-section of DNA because this one looks slightly different to the one I showed in the previous slide, and sometimes in exams they will try and trip you up. They'll make it so that the directionality is not as easy to infer, and sometimes they won't even label the nitrogenous bases. You have to use your kind of knowledge of the chemistry and the structure of both purines and both pyrimidines to kind of infer what nitrogenous base is what and what the directionality is. So I'm going to use this two-dimensional cross-section to help everybody understand how to do that. But before we do that, just to kind of reiterate, the hydrogen bonding that you see in the DNA double helix occurs between the nitrogenous bases and their respective functional groups. So just to kind of break it down, we'll start with the top one. You can see that adenine thymine hydrogen bond relationship, and one way you can kind of infer is that lack of that kind of third hydrogen bonding, which is indicated by no line. So you can see 2 lines on the adenine thymine and 3 on the guanine cytosine. The guanine cytosine bond, because of the functional grouping, is much more stable. It's much more thermodynamically stable. So you can see that adenine thymine has only 2 hydrogen bonds and guanine and cytosine has 3. So that's the first kind of clue. Okay, and you also know as well that adenine has that amine group on its 6th carbon, whereas guanine has that carbonyl functional group, and that amine group is that NH2. And in terms of how are we distinguishing purines from pyrimidines? Well, we know that purines have 2 heterocyclic rings. They've got 2 heterocyclic aromatic rings. One's an imidazole ring, that 5-membered ring that will bond with that deoxyribose sugar through a glycosidic bond, and then the other is that pyrimidine 6-membered ring, whereas pyrimidines like thymine and cytosine only have that 6-membered ring. But anyway, going back to kind of like understanding this two-dimensional cross-section, if you look at adenine and thymine, you can see that that carbonyl group on the thymine is hydrogen bonded with that amine group on the adenine. So that oxygen on that carbonyl group is acting as the hydrogen acceptor. Okay, and we know that because oxygen is going to be electronegative. It's going to have that electronegative symbol. If you're looking at the carbonyl functional group, it's going to be holding those electrons because of shielding, and because of that atomic nuclei exposure, less shielding than carbon, is going to be pulling those electrons. Okay, and conversely, that amine group, nitrogen's a very, a larger electronegative atom than hydrogen is. There's more pull there. Okay, so that hydrogen in an amine group will always be electropositive, and it will always have the capability of acting as a hydrogen donor. So there's a weak intermolecular force that's formed between that carbonyl group and that amine, that carbonyl group on the thymine pyrimidine, and that amine group on the adenine purine. Now, if we go down, you can see the guanine cytosine. Okay, there's three, the guanine cytosine complementary pairing. There's 3 hydrogen bonds there overall. Okay, and that makes it more thermodynamically stable. More energy is going to be required to break those bonds, and that's a really important concept when we go into kind of like the mechanisms behind DNA replication. So you can see there, first of all, that guanine again is a purine. It's got that imidazole ring that's linked to the deoxyribose sugar through that glycosidic bond, that nitrogen bridge, and then you've got that 6-membered pyrimidine ring, again, still a heterocyclic ring. And guanine has that carbonyl group on its 6th carbon, and it also has that kind of additional amine functional group that adenine doesn't have. Adenine's only got, kind of one textbook functional group, I guess. And cytosine has that carbonyl group and that amine group. It lacks that methyl group. Thymine does not have that amine group, but it has that methyl group. If you remember as well, uracil is that other nitrogenous pyrimidine base that's not incorporated in DNA. It's present in RNA though, which I'll go through in another video in protein synthesis when we look at transcription and translation. But uracil is pretty structurally similar to thymine. It lacks that kind of methyl group. And I know that if a lot of you, if you know your kind of metabolism and biochemistry, where uracil, thymine can be synthesized from uracil using the enzyme thymidylate synthase, and I think folate as a cofactor, and adding that methyl group gives you thymine. Now we're going to talk more about the directionality, because I think it's harder to infer directionality in diagrams like this, and examiners will purposely give you this to kind of throw you off. In the previous one, the functional groups were easily inferred, but in this one you kind of have to use your knowledge of the phosphate sugar backbone pattern, that kind of 5,3,5,3 hydroxyl phosphate pattern, to kind of infer the directionality. So my head's covering the right hand side, so I'll use the left hand side as an example. So you can see on the left hand side at the top, you've got what's the 3' terminal. Okay, and typically there should be a hydroxyl group, not that phosphate group. A lot of people get confused because that phosphate, as you know, is bonded to that 5th carbon. Okay, but it's not, because it is bonded to that 5th carbon, but you can see that the polymer chain has not actually ended. There's another line or arrow indicating that kind of bond angle, indicating there's an upstream deoxyribose sugar. Okay, so that must mean that phosphate would be bonded to that deoxyribose sugar, okay, on that 5th carbon, leaving that 3rd carbon, that hydroxyl group on the 3rd carbon, exposed at the N terminal. Now, you could maybe argue that there could be another kind of phosphate sugar after that, but we don't know that. All we know is that what we're given there. So we would just assume that would be the 3' direction, and that must mean the other side would be the 5' direction. Okay, so that's just a bit of background on how to kind of predict the directionality, and that's due to the kind of functional groups that are sticking out of the kind of N terminal of the polynucleotide. So now we're going to be looking at DNA replication, and it's a 3-process mechanism. We've got initiation, then you've got elongation, then you've got termination. So I'll go through all of them, and we're going to talk about all the key enzymes involved, all the kind of mediator signaling proteins involved in this process. So what is DNA replication? So DNA replication is a process in which a cell replicates its entire genome and produces two identical DNA molecules. So one DNA helix produces two daughter helices. Now it's said to be semi-conservative in nature, okay, and this means that each of those helices, each of those polynucleotide strands in the original DNA helix will create two daughter helices. Those two parent helices will be used as a template helix for our daughter helices to be formed. So each of the new identical daughter helices that are formed contains one strand from the parent strand and one new daughter helix altogether. Okay, so and that kind of speaks to its semi-conservative nature. Now, DNA is also synthesized in what's called a complementary manner. So it's synthesized, it's polymerized in a 5 to 3' direction. And as you know, that's really important. The chemistry of DNA, it has to be synthesized in that direction because we need that hydroxyl group to act as a nucleotide for our incoming nucleotide triphosphates, okay, to kind of form that phosphodiester bond that kind of bridges that phosphate sugar backbone together. Now, but the template strands, those parent strands that I mentioned before in the parent helix that are used to form our daughter strands, they're read in a 3 to 5' direction. So again, this speaks of the directionality of DNA, that those functional groups, the phosphate and hydroxyl groups, and what carbon they're bonded to on the deoxyribose sugar. So it's read in a 3 to 5' direction. So it's semi-conservative in nature. That's a really important point, and it's read in a 3 to 5' direction, and it's synthesized. The daughter strand is synthesized from the parent strands in a 5 to 3' direction. Now, why does DNA replication occur? So DNA replication occurs before mitosis happens. And as you know, mitosis is a process in which a cell will divide and form two genetically identical daughter cells. Okay, and and again, that speaks to the kind of integrity of DNA replication. Okay, it ensures that the genetic information that's being passed down is faithful to that of the parent cell. Those daughter cells possess the same genetic sequence as the parent cells. Now, DNA replication mainly occurs in the S phase of the cell cycle, but it's actually initiated in the G1 phase of the cell cycle. That pre-replication protein complex is assembled in the G1 phase of the cell cycle, and it's assembled in its inactive state. And then once that pre-replication protein complex is activated by our cyclin-dependent and our D-dependent kinase enzymes, okay, we kind of move into the S phase of the cell cycle, and DNA replication is initiated. Now, a lot of you might confuse mitosis with meiosis. Now, I just want to make it clear that mitosis is a very different process to meiosis. Meiosis refers to more sexual reproduction, okay, and that's what a cell will divide and form 4 genetically different daughter cells, which I'll go over in another video. But we're talking about mitosis here. We're just briefly kind of touching on it to kind of give a bit of background as to why DNA replication is so important. Before a cell divides, it will replicate its entire genome. Okay, and just to recap, if the main takeaway from this slide is DNA itself is the DNA replication itself is semi-conservative in nature. So as I said before, that original helix will form two daughter helices, and those daughter helices will contain one strand, one polynucleotide strand from the parent helix, and that is used as a template strand for our enzyme, DNA polymerase, to form a new daughter strand, okay, and that daughter strand is polymerized in a 5 to 3' fashion, okay. That 3' hydroxyl groups act as a nucleophile for our incoming nucleotide triphosphates, okay, but that DNA polymerase is reading our parent template strand in a 3 to 5' direction, okay. And if you kind of understand the directionality of DNA, like we said before, DNA is anti-parallel in nature, which means the replication process is going to be bidirectional, because our DNA polymerase can only read the strand in a 3 to 5' direction. Our primase enzymes can only read the strand in a 3 to 5' direction. And because of the chemistry of DNA polymerization, or nucleic acid polymerization, we know that has to happen in a 5 to 3' direction. So because of that, it's bidirectional in nature. So it's semi-conservative, it's anti-parallel, and it's bidirectional. So again, I just kind of want to reinforce this kind of concept of DNA polymerization, okay, and the kind of chemistry behind it again. We touched on this in the last video, or we went over it in the last video, okay, but I'm going to now go over it again and kind of apply it to our kind of DNA replication, just so we can build a picture of what's happening on a kind of molecular level, if you like. So you can see the equation I've got on the top is the DNA NOH plus the DNTP, and all this means is we've got our growing DNA, we've got a DNA polymer, okay, the N just is an algebraic expression to refer to the fact that we don't know how many nucleotides are making up this polymer, just enough to be considered a polynucleotide, okay, and that OH group, that hydroxyl group, is that 3', okay, that third carbon on the deoxyribose sugar, that exposed hydroxyl group is acting as a nucleophile to the incoming deoxynucleotide triphosphates. Okay, so it's allowing for that polymerization process to happen. Okay, so it's integral that we have that OH group, that hydroxyl group, that 3' exposed at the terminal of our growing polymer at all times. Okay, otherwise the polymerization process can't happen. Okay, because it's the chemistry. The hydroxyl group acts as a nucleophile. Okay, the phosphoanhydride bond that links the beta and alpha phosphates is cleaved on the incoming deoxynucleotide triphosphate. Okay, that provides the energy, the Gibbs energy. Okay, that free energy that allows the work to happen. That kind of allows that polymerization process to happen. We get pyrophosphate or diphosphate released as a byproduct. Okay, and that's just the beta and gamma phosphates being cleaved off, okay, and that phosphate, which is attached to the 5th carbon of the deoxyribose sugar, will form a phosphoester linkage, or in this case a phosphodiester linkage, with that hydroxyl group on the 3rd carbon. So therefore, the way it would orientate in leaving that incoming deoxynucleotide triphosphate, that one that we've just bonded, okay, with the DNA NOH, okay, leaving the incoming DNTP, that 3rd carbon, exposed on the deoxyribose sugar again. So we've got a hydroxyl group is exposed again, which is important. We need that to act as a nucleophile and to form that phosphate sugar linkage, that 5'3" kind of linkage, if you like. So that's why it's so important to kind of understand the directionality of DNA, okay? So it can only therefore be synthesized in a 5'3" direction because of the chemistry, because of orientation, because of the functional groups, and the way in which DNA will polymerize together, okay? So therefore, because of the antiparallel nature of DNA, if it's forming a daughter strand from a template parent strand, okay, and if the polymerization process is happening in a 5'3" direction, the strand must be read in a 3' to 5' direction, okay? So DNA polymerase is not only reading the parent template strand in a 5'3" direction, it's actually forming a new daughter strand in a 5'3" fashion, okay? So it's reading the template in a 3 to 5 direction and forming the daughter strand in a 5'3" fashion. So it's a very, very clever enzyme, if you like, okay? And again, just to kind of reiterate to avoid confusion, we have incoming nucleotide triphosphates, and we got there through stepwise phosphorylation, which I went over in the previous video, okay? And those phosphoanhydride bonds that kind of link those those beta, alpha, and gamma phosphates together, okay, when they're broken, a lot of chemical potential energy is released, okay, which is driving this process, okay? And again, just to reinforce, magnesium is acting as a charge stabilizer, okay, so it's acting as a shield, if you like, preventing that natural electrostatic repulsion that will occur between a negatively charged hydroxyl group and a negatively charged phosphate group, okay. So it's acting as a charge shield. It's stabilizing that enzyme substrate complex by reducing electrostatic repulsion. Okay, and again it's referred to as a phosphodiester bond. Okay, because that phosphate group has attached to the 3rd carbon of the deoxyribose sugar. Okay, but that phosphate group is located on the 5th carbon, and it's so that it will therefore have the ability, that sugar, deoxyribose sugar, will have the ability on its exposed hydroxyl group, okay, and its 3rd carbon, okay, its 3rd carbon terminal will be able to form another bond with an incoming deoxynucleotide triphosphate. Okay, I know it's confusing, but if you can build that picture in your head, it allows understanding this process so much easier, okay, and it's that phosphate sugar linkage that allows for the polymerization to happen.
So just to kind of recap everything we've spoken about, we'll start from the beginning. So DNA is a helical structure as it sits in cells. Its canonical form is a helix, which consists of 2 polynucleotides or polymers of DNA kind of wrapped around each other. They're not actually touching, and that helical structure is attributed to a complementary pairing between our nitrogenous bases, our purines and our pyrimidines, and that relies on hydrogen bonding, okay, those weak intermolecular forces between hydrogen on a functional group of 1 molecule and an electronegative atom on a functional group on a neighboring molecule. Those intermolecular forces, those complementary pairing, kind of govern the DNA's helical structure. Now, DNA itself is anti-parallel in nature, so those two helices, as they're wrapped around each other, the directionality is different on both strands because of the phosphate backbone. Whatever functional group is sticking out of that kind of N terminal will essentially be it will essentially dictate its directionality, if you like. So we know that one strand will run in a 3 to 5 prime direction and the other on a 5 to 3 prime direction. Okay, and what do we mean by 3 prime? Well, 3 prime just refers to that hydroxyl group on that third carbon, okay, on that deoxyribose sugar that's kind of partially making up that phosphate sugar backbone, okay, which means conversely that phosphate group is bonded to that fifth carbon on the deoxyribose sugar, and that will form a linkage, a phosphodiester linkage downstream or upstream with another deoxyribose sugar, okay, on that third carbon, that hydroxyl group, kind of acting as a nucleophile, okay, and that concept, that pattern, that phosphate sugar is critical in not only understanding the directionality of DNA itself, but also the kind of chemistry behind DNA polymerization. Now, DNA replication itself, okay, is semi-conservative in nature. Okay, so what does that mean? It means that one DNA helix will form two daughter helices. Each of those daughter helices is not only genetically identical to the parent helix, but each daughter helix contains one strand from the parent helix, and that strand was used as a template strand for DNA. In each helix, that parent strand was used as a template strand for DNA polymerase to read that template strand in a 3 to 5 prime direction and synthesize its daughter strand in a 5 to 3 prime direction. Okay, and why are we synthesizing it in a 5 to 3 prime direction? Because we need that 3 prime hydroxyl group to act as a nucleophile for our incoming deoxynucleotide triphosphates, and we essentially that process is critical in nucleic acid polymerization. And because of DNA's anti-parallel nature, it means the replication process is naturally going to be bidirectional, because our enzyme can only read, our DNA polymerase, our primase enzymes, they can only read the template strands in a 3 to 5 prime fashion, and DNA itself, nucleic acids itself, can only be formed or synthesized in a 5 to 3 prime fashion because of that hydroxyl group we need. And again, just to quickly recap the concept of DNA polymerization for those of you who are still confused, that hydroxyl group is critical in acting as a nucleophile. Okay, those incoming deoxynucleotide triphosphates, they contain that beta and that gamma phosphate that will eventually be cleaved off before the polymerization process. Okay, and that allows for that energy released from that phosphoanhydride bond that's stored, that chemical potential energy that's stored in that beta and gamma phosphate on our incoming deoxynucleotide triphosphate, that formation of pyrophosphate and the subsequent release of that chemical potential energy provides that work, the energy for that work to happen and for that bond to form. Okay, and our growing DNA strand, okay, our daughter strands are formed in that kind of 5 to 3 prime fashion, and our parent strands are read in that 3 to 5 prime fashion. So so that they're the kind of concepts, I guess, that are really important so far to kind of understand, or to begin to understand, the DNA replication process. So I've got a diagram here showing what's called a replication bubble, or replication fork, if you like. Now, this may seem, to a lot of people who don't understand this process, it may look quite daunting. You might not understand what's going on, but as we progress through the videos, this process will become a lot clearer. But I thought I'd kind of summarize this diagram before we kind of move on, to kind of help people understand these kind of key terms like bidirectionality, like semi-conservative nature, anti-parallel nature. So to start off with, in blue, you've got your parent strands, okay? So as we know, DNA replication is a process in which you have one original helix, okay, and you form two identical daughter helices. And both those daughter helices contain one strand from the original parent strand, okay, and those strands were used as template strands for our enzymes like DNA polymerase and primase to DNA polymerase mainly, and primase to kind of assist and help build that daughter strand. Okay, and that daughter strand is formed in a 5 to 3 prime fashion because of the chemistry behind nucleic acid polymerization, and that template strand is naturally read in a 3 to 5 prime fashion. And we know that DNA is anti-parallel in nature, so one strand's going to be running in a 5 to 3 prime direction, the other naturally will be running in a 3 to 5 prime direction, which means that the process itself is bidirectional. So our DNA polymerase for each parent strand will be going in a different direction. Now, so as I said, in blue, you've got your kind of parent strands, and they're being used as template strands. And in red, you've got your daughter strands. Green are RNA primers. Now, RNA primers are used to kind of initiate the process of DNA polymerization. Now, I'll go through RNA a lot when we look at protein synthesis, but for now, I guess all you need to know is RNA primers, they're synthesized by the primase enzyme. Now, this is a kind of concept that I'll get into more as we move on, but RNA primers can be synthesized de novo, which means from scratch, whereas DNA can't be synthesized de novo from scratch. Okay, DNA requires the use of RNA primers, and then that daughter strand can use those primers to kind of initiate and build upon its daughter strand. And the reason for that being, okay, is that you need that hydroxyl group to be exposed. And that, though, for the chemistry, for the polymerization process to happen, and that exposure of the hydroxyl group on the RNA primer on the 3' end of the primer terminal, if you like, can act as that nucleophile, and then you can have an incoming deoxynucleotide triphosphate to initiate that growing strand, that daughter strand. Now, something that I think I might have missed in the previous slides that I'll just kind of reiterate is that the DNA polymerase is forming its daughter strand in a complementary fashion. So it's reading the template strand, okay, and let's say it's reading adenine, it will then incorporate a thymine. If it's reading cytosine, it will incorporate a guanine. So it's that kind of it's retaining that complementary sequencing, which is integral in ensuring the genetic information that's passed down or is faithful as the cell divides. Now, you can see that the top strand is referred to as a leading strand. Now, this is moving towards the replication fork, if you like, so it allows for a continuous synthesis of DNA once the RNA primer has been put there, synthesized de novo. It allows a DNA polymerase to then continuously synthesize its daughter strand, okay, reading it in a 3 to 5' direction and synthesizing or assembling that daughter strand in a 5 to 3' fashion, okay. But the lagging strand is a little bit different. So the lagging strand requires multiple RNA primers strategically placed across opposite or parallel to the template strand, and then DNA polymerase will discontinuously synthesize these kind of short DNA fragments in between these RNA primers, and they're called Okazaki fragments. And once those primers are removed by RNase H and Fen1, and those enzymes associated with primer removal, okay, then delta polymerase will come in and refill those gaps, okay, and then DNA ligase will seal that daughter strand on the lagging strand, will seal that phosphate backbone. And I'll get into all of this as we move through the slides. Now, and that's what those Okazaki fragments are. They refer to those kind of discontinuous fragments of DNA between those RNA primers. But essentially, that's the kind of initiation and elongation process. And you could see helicases, those kind of blue triangles, and they're running in a bidirectional manner again. And what they're doing is they're breaking those hydrogen bonds between the two parent strands. So those hydrogen bonds, that helical structure is being broken, and it's forming these kind of bubbles, and those parent templates are being exposed, and they're being used as it's allowing them to be used as templates for our enzymes like DNA polymerase, primase, or delta polymerase, DNA polymerase, primase to kind of run across, read those parent strands, and put the appropriate or incorporate the appropriate nucleotide base depending on what the template strand is reading. So that's essentially that whole process, and we'll talk about it more in the slides to come. So as I said before, there are 3 stages to DNA replication. The first stage is the initiation phase, and this starts off in the G1 phase of the cell cycle. Okay, as I mentioned before, the G1 phase of the cell cycle is where we see our pre-replication protein assembly, okay, that form that kind of pre-replication protein complex. Okay, so those protein constituents assemble to form this pre-replication protein complex, and the activation of this protein complex essentially marks that transition from G1 phase into the S phase of the cell cycle, or the synthesis phase, and that's where we start to get our actual DNA synthesis. Okay, now in the G1 phase of the cell cycle, you get the assembly of the pre-replication protein complex. And the pre-replication protein complex is a multi-protein complex made up of several different protein constituents. Now, they will bind and form these kind of loading platforms to allow our helicase enzymes to offload bidirectionally and kind of split that double helix structure. Now, I thought I'd talk about this a bit more because there's a lot of conflation in literature, just to kind of help people understand what the pre-replication protein complex actually does. So the first constituent we're going to talk about is the ORC complex, okay, and that's the origin recognition complex. And that's a protein complex or a constituent of the pre-replication protein complex that's able to recognize the origin of replication sites. Now, the origin of replication sites refer to these adenine thymine rich areas in a DNA double helix, and there's multiple origin of replication sites or replication sites in the DNA double helix because it's a very long helical structure. Now, as I said before, the adenine thymine complementary pairing, that purine adenine and that pyrimine and thymine, that complementary pairing only has 2 hydrogen bonds compared to cytosine and guanine, so making it less thermodynamically stable. As we know, guanine and cytosine contribute massively towards the kind of thermodynamic stability of DNA, whereas adenine and thymine, they only have those 2 hydrogen bonds. And as you know, that complementary pairing is consistent across the helical structure. So if you look at the origin of replication site, if you look at take one of those parent strands out, you'll see multiple adenine sequencing back to back. So A. Okay, that means conversely the other strand, the other strand is going to have lots of thymine sequencing. So T. Okay, and those lots a lot of adenine thymine complementary pairing consistently across the origin of replication site creates these kind of more or less thermodynamically stable portions of the double helix to allow our helicases to kind of have a head start once they offload from this kind of pre-replication protein complex. So the origin recognition complex is the first constituent of the pre-replication protein complex, and the origin recognition complex will recognize, it will bind to those origin of replication sites, so it binds to those adenine thymine rich areas. But it's important to remember that our DNA double helix is still intact at this point. DNA still retained its helical structure. Okay, we're in a G1 phase of the cell cycle. The ORC complex is bound to that double helix, it's bound to those origin of replication sites, those adenine thymine rich areas, and the helical structure has been retained. Now, shortly after binding to the origin of replication sites, 2 other protein constituents will load onto the ORC complex, and that's a CDC6 constituent, okay, and the CDT1 constituent. So the CDC6 constituent will load on first, and then the CDT1 constituent will load on next to it, okay, and it forms this kind of loading platform for our MCM27 dimer, okay, and that our MCM27 dimer to load on to sit on top of that, and by MCM27 dimer I mean our MCM27 helicase complex. So there's 2 hexameric helicases are loaded back to back, okay, and that allows for that kind of bidirectional travel of helicase because it's this kind of active motor protein. It allows it to kind of offload and split the double helix in either direction, okay, exposing both those kind of template strands. Now, when the CD6 complex binds to the ORC DNA complex, that requires ATP, okay, and then that kind of results in a conformational change of the CDC6 constituent, and that allows for the CDT1 to kind of load on top of that, and it's the loading of the CDT1 that allows for the MCM27 dimer to be loaded on top of that, and it's essentially like this kind of loading platform for our helicase enzymes to kind of offload and start breaking that double helix. So I just thought I'd kind of touch more on this kind of concept of the pre-replication complex because I think there's a lot of conflation in literature, as I said before. So the pre-replication complex doesn't actually break those hydrogen bonds. It doesn't split that strand of DNA, that parent strand. What it does is it acts as a loading platform and a layer of regulation. Okay, now it acts as a loading platform for our MCM27 dimers, okay, and that's just our 2 helicases loaded back to back to offload, become our active CMG helicases and break those hydrogen bonds. So the helicase is still breaking those hydrogen bonds, but the pre-replication protein complex is acting as a loading platform to position our helicases, give them a head start because of that, to recognize those less thermodynamically stable areas or portions of the double helix because of that adenine thymine rich pairing, and it adds a layer of regulation as well because which I'll talk about in the coming slides. Our cyclin dependent kinase and our D dependent kinase will the D dependent kinase will activate those those helicases, okay, allow them to undergo their conformational change and recruit their respective protein constituents to become active CMG helicases. And then our cyclin-dependent kinase will phosphorylate the CDT1 and CDC6 platform to prevent the relicensing of another helicase. So it adds a layer of regulation. This pre-replication protein complex adds a layer of regulation as well. Now, the ORC DNA interactions or ORC DNA binding is stabilized by the kind of hydrogen bonds you see in those exposed functional groups between adenine and thymine. Okay, and it also hydrogen bonds or forms these kind of electrostatic forces, if you like, with the phosphate backbone itself. So these adenine thymine rich areas favor ORC docking. Okay, and that ORC, that origin recognition complex, that protein constituent, is integral in initiating the kind of assembly of the pre-replication protein complex. And then that pre-replication protein complex is essentially in kind of regulating helicase activity and preventing relicensing of those MC-M27 dimers back onto the CDT1 and CDC6 kind of loading platforms. So I just thought I'd show this diagram to kind of reiterate and emphasize what's actually going on with the pre-replication protein complex and what role it plays in DNA replication. As I said before, you've got the G1 phase, okay, and as we know, DNA replication predominantly occurs, or pretty much exclusively occurs in the S phase, but the G1 phase marks the kind of assembly of the inactive pre-replication protein complex, okay, and that's that multi-protein complex, and those constituents will kind of assemble together in this kind of sequence. So the ORC complex, or the origin recognition complex, is the first protein constituent to bind to the DNA. Okay, is that kind of initiator in the formation of this pre-replication protein complex. Okay, and the ORC complex, that functions as a kind of like a sensor. It can kind of recognize those origin of replication sites, and as we know, on the DNA double helix, you've got multiple origin of replication sites. They're the adenine thymine rich areas. They're rich in adenine thymine complementary base pairing, and as we know, that means it's less thermodynamically stable because of the hydrogen bonding that occurs between adenine and thymine. Okay, so it's giving our helicases a head start eventually when they will offload and break that double helix, okay, to expose those template strands. So once the ORC complex is bound to the origin of recognition site, shortly after, our CDC6 and our CDT1 constituents will follow. Now, our CDC6 constituent follows first with the help of ATP. That phosphate coupling results in a conformational change, and our CDT1 can then kind of assemble next to that and forming this kind of loading platform for our MC-M27 dimer to load on top. And our MC-M27 dimer refers to our two inactive helicases that are loaded back to back in this protein dimer. Okay, and that will eventually allow for a bidirectional travel. They'll offload and they'll go bidirectionally, one in one direction, one in the other, to split that double helix open. Now, in the G1 phase of the cell cycle, we have our ORC complex, our CDC6 complex, our CDT1 complex, and our MC-M27 dimer, all loaded in this kind of protein complex. Now, as we move to the S phase of the cell cycle, we have two enzymes, our DBF4 dependent kinase, or we'll call it D-dependent kinase, and our CDK, or cyclin dependent kinase, and they're both kinase enzymes, and they're both involved in kind of phosphorylating this protein complex, but they play slightly different roles. So the D-dependent kinase is a really important enzyme in activating our helicase enzymes. Okay, so we go from a MC-M27 protein to an active CMG helicase. So once D-dependent kinase phosphorylates this MC-M27 dimer, what happens is each of those MC-M27 constituents that make up this MC-M27 dimer will recruit respectively a CDC45 and a GINS protein constituent. Okay, and that phosphate coupling causes that conformational change. It causes that protein recruitment, allows for those constituents to bind to our MC-M27. Okay, and that will form our CMG helicase. So each helicase enzyme, each hexameric helicase, has now got a CDC45 and a GINS constituent bound to it, and that allows it to kind of offload and become this kind of active motor protein and break those hydrogen bonds. Now, once our helicases are offloaded, cyclin-dependent kinase will phosphorylate the CDC6 and CDT1 loading platform, and the OCR Okay, and that prevents the relicensing of another MCM27 dimer, and that's a really crucial step. That adds a kind of layer of regulation, if you like, because we've already got our helicases running across to break that strand. We don't need another lot of helicases offloading. Okay, and once cyclin-dependent kinase phosphorylates those remaining constituents, they disassemble and they're degraded, okay, in the metabolism, which we won't really get into. It's not really that important, but the main concept to kind of understand is the G1 phase is that kind of assembly of this pre-replication protein complex, okay, which consists of that ORC constituent, the CDC6 and the CDT1 constituents, that once CDC6 is kind of phosphorylated, it undergoes that conformational change. It allows for the recruitment of the CDT1 constituent that acts as a loading platform for our MCM27 dimer, and our MCM27 dimer is just 2 hexameric helicases loaded back to back to allow for that kind of bidirectional travel, okay, and then the S phase marks the activation of this pre-replication protein complex. So D-dependent kinase will phosphorylate the MCM27 dimer. That results in a conformational change, that phosphate coupling, and each of those helicase enzymes will recruit their CDC4/5 and GINS constituents respectively, and they become active CMG helicases. Okay, they're motor proteins that will break that bond. Okay, and then those hydrogen bonds, that complementary pairing between those two helices in the DNA double helix, allowing for those parent template strands to be exposed for our DNA polymerase and our RNA or RNA primase to kind of work on those template strands. And then cyclin-dependent kinase will come and phosphorylate the remaining CDC6 and CDT1 constituents to prevent the relicensing of another MCM27 dimer. And that's essentially this kind of like the role that this pre-replication process plays in kind of initiating or initiating our initiation phase of DNA replication. Okay, and that initial ORC, origin of recognition complex binding, was really important in recognizing those areas that are less thermodynamically stable on the DNA double helix. It recognizes those adenine thymine rich like complementary pairing areas. Now, once, as I said before, that binding of the pre-replication protein complex isn't necessarily breaking those hydrogen bonds. So in the G1 phase, you can have that pre-replication protein complex assembled and bound to your DNA double helix, but it's important to remember that helical structure is still retained. It's only disrupted. You only get your replication bubble. You only get the break of the bonds when D-dependent kinase comes and phosphorylates the MCM27 dimer. Okay, respectively, a D-dependent kinase will phosphorylate each of those hexameric helicases that are loaded back to back in that protein dimer to allow for the respective recruitment of those CDC4/5 and GINS constituents. Okay, and once they've coupled with those constituents, they become active motor proteins. So, and it's those active motor proteins, that helicase enzyme, our CMG helicases, that actually breaking those hydrogen bonds, but they've been given a head start because the areas they're breaking through first are less thermodynamically stable. Those adenine thymine rich areas, it's kind of giving them a head start, if you like. And obviously our cyclin-dependent kinase will come and phosphorylate the remaining CDC6 and CDC1 constituents to kind of prevent that relicensing. It adds that layer of regulation to this process. Just to kind of recap on the role of these kinase enzymes in this process. So the cyclin-dependent kinase phosphorylates the ORC, CD6 and CDT1 constituents, preventing the relicensing of another MCM helicase complex, okay, or an MCM27 dimer. And our D-dependent kinase will directly phosphorylate our helicases to allow them, this kind of phosphate coupling allows them to undergo that conformational change, and they can recruit and bind their active or their respective GINS and CDC4/5 constituents. And that when helicase is when it's bound to those constituents, that GINS and CDC4/5 is considered an active motor protein. Okay, so during the G1 phase of the cell cycle, our MCM27 dimers they're not bound. They haven't that GINS or CDC4/5 constituents are yet to be incorporated into their structure. Okay, they're just those MCM27 dimers. It's in the S phase of the cell cycle after D-dependent kinases that has phosphorylated the MCM27 dimer. Okay, that phosphate coupling allows for that recruitment of the GINS and CDC4/5 constituents, and that's when they become active motor proteins, and that's in the S phase of the cell cycle. So just to kind of recap, CDK or cyclin-dependent kinase prevents the relicensing of another MCM27 helicase complex or MCM27 dimer, and it does that by phosphorylating the ORC, CD6 and CDT1 platforms, which remain after D-dependent kinases phosphorylated our MCM27 complex to allow that offloading, okay, and cyclin dependent kinases phosphorylated those remaining protein constituents to prevent that relicensing, okay, and that those remaining constituents, CDC6, the CDT1 and the ORC, will disassemble and they'll get degraded, okay, while our helicases are still active motor proteins running along and breaking those hydrogen bonds in that kind of double helix, exposing those template strands to allow our enzymes to start building our respective daughter strands.
So now we're going to talk about the single-stranded binding protein. So we've just spoken about the assembly of the pre-replication protein complex, its activation via phosphorylation of two specific enzymes, cyclin-dependent kinase and D-dependent kinase. So now we've got our active helicases, or our CMG helicases, running in a bidirectional manner, splitting that double strand, breaking those hydrogen bonds. And as we form our replication fork, if you like, a special type of protein is recruited. It's referred to as single-stranded binding protein, or SSB in the diagram, and it binds to that phosphate backbone. And what that does is it prevents the reannealing of DNA. So as it's being split, those hydrogen bonds are going to want to reform, and it has a propensity to want to reform into its canonical helical structure, okay, and single-stranded binding protein just prevents this from happening, okay, allowing for those two parent strands to act as templates, okay, as they're being separated. It also protects the backbone, the phosphate sugar backbone, from certain nuclease enzymes, which typically will hydrolyze phosphodiester bonds downstream, where the double helix is still intact, in order to relieve that supercoiling. But to make sure it doesn't affect the template strands, the single-stranded binding protein will bind on that phosphate sugar backbone. So I just thought I'd demonstrate as well, just a bit of background on what these helicases look like, and just to kind of demystify if anyone's confused. So when I say the MCM27 dimers, I mean the two hexameric structures are loaded back to back, so the two inactive helicases are loaded back to back on the loading platform, on the CDC6 CDT1 loading platform. And then when they're phosphorylated by D-dependent kinase, both of them will respectively recruit those constituents GINS and CDC45, and that's when they become active, and they go in a kind of bidirectional manner, and they run across that double helix strand. The helicases themselves, we call them hexameric because they've got 6 protein subunits, okay, and those subunits form what's called like a toroidal ring, so it looks like a donut structure. You can see the blue ring-like structure is composed of the 6 protein units, and it's in that kind of donut shape. And that's just the helicase enzyme, and it's referred to as an MCM27 when it's inactive, and it becomes CMG helicase when it's activated. So a few slides ago I spoke about the single-stranded binding protein and how it binds to that phosphate sugar backbone once helicase has split that strand and created that replication fork. Downstream from that split, though, we get what's called supercoiling or torsonal strain. So the helical structure becomes distorted, okay, and helicase is essentially working against itself. So those enzymes that are breaking the strand, eventually, without this enzyme, without topoisomerase 1, essentially what's going to happen is they're going to come to quite a quick standstill, because the supercoiling in the DNA will prevent them from running across smoothly and breaking those hydrogen bonds. So topoisomerase type 1 works downstream from, or upstream, depending on what direction you're looking at the helicases, because remember the helicases are traveling in what's called a bidirectional manner, okay, so either upstream or downstream from where those helicases are traveling, where the double helix is still intact, where that helical structure is retained, topoisomerase 1 relieves that supercoiling, if you like, allowing for helicase to continue, okay, and those single-stranded binding proteins protect our template strands once they're split open from enzymes like this. Okay, so a lot of people might be familiar with topoisomerase 1. It's a target for a lot of therapies, both antimicrobial, okay, and cancer therapies as well, to prevent cell replication. But to talk about this enzyme, okay, so there's topoisomerase type 1, and then there's topoisomerase type 2A, which we're going to talk about next, which is more involved after the replication process. Topoisomerase type 1 itself only has one functional domain, so one nuclease domain, and in its active site it contains the amino acid tyrosine.
tyrosine in its R group, as a lot of people should be familiar with, contains what's called a phenol ring or an alcohol ring, so it's got that hydroxyl group. And I'll go over amino acids in a few videos time, but for now all you need to know is that phenol ring allows for a transesterification reaction between the phosphate backbone, okay, and the enzyme itself. So that active site, that hydroxyl group, is going to act as a nucleophile, and it's going to steal that phosphate group, okay, from the backbone, okay, forming a free phosphotyrosal intermediate, okay, and that's referred to as transesterification. And then what's going to happen is the nucleotide, the hydroxyl group that's on the 5th carbon of the nucleotide downstream, so nucleotide B, will want that bond back. Okay, so the bond will, the ester bond will quickly reform in the phosphate sugar backbone, but as it's taken that bond back, it causes this kind of rotation, and that rotation is what relieves the supercoiling or the torsonal strain. So that's how topoisomerase relieves type 1 relieves strain of the DNA double helix, okay, for a transesterification reaction, temporarily breaking that backbone in an ATP independent manner, okay, and then that rotation on a downstream nucleotide or upstream, depending on what way you're looking at it, will result in that relief in tangling, and you get that religation in the backbone. Now, topoisomerase, just to kind of emphasize, it's an ATP independent enzyme, topoisomerase type 1. Topoisomerase type 2A, which we'll talk about next, is an ATP dependent enzyme. So the next enzyme we're going to talk about is the topoisomerase type 2A enzyme. Now that's different to the topoisomerase type 1. This is a homodimer enzyme, so it contains 2 subunits, okay? And both those subunits are nuclease subunits, the same as topoisomerase type 1. Now it's important we refer to topoisomerase type 2A as topoisomerase type 2A because topoisomerase type 2 is the topoisomerase that's present in prokaryotic or single-celled organisms like bacteria. Us being eukaryotes or multicellular organisms, ours is referred to as type 2A. And essentially this topoisomerase type 2A plays a really important role in the termination phase of DNA replication. So it what calls decatenates the DNA, so it untangles the 2 daughter helices once they formed. Once you get those 2 helices after the initiation and elongation during DNA replication, you get the 2 daughter helices in the termination phase, and they're kind of tangled across each other. And topoisomerase type 2A just works by untangling those 2 helices, and that's why it has 2 domains, so it can bind to the backbone of both helices. And it forms what's called a 5' phosphotyrosal intermediate. So again, it's got tyrosine in its active site, and that phenol ring, that hydroxyl group on the phenol ring, is acting as a nucleophile. It's taking that, and then obviously that 3' hydroxyl group on the downstream nucleotide is going to want that bond back. So it's going to religate, and that religation causes rotation, and that rotation essentially relieves that tangling. Okay, this is an ATP dependent enzyme, so it requires ATP to work, not to break the bonds, but it requires phosphate coupling to result in that conformational change that will allow it to become active. So I'm just going to recap the initiation process that we just kind of spoke about, just to reinforce these concepts. So we start off in the G1 phase of our cell cycle, and that's when we essentially assemble the inactive form of our pre-replication recognition protein. Okay, so that's a multi-protein complex made up of several constituents. Okay, the first constituent is the ORC, or the origin recognition complex, okay, and that recognizes the replication origin sites on the DNA double helix. So they're those adenine thymine dense areas, okay, and it's those areas. Why do we choose those areas? Because they're less thermodynamically stable, okay. There's less hydrogen bonding there overall because there's less hydrogen bonding between the adenine and thymine nitrogenous bases, okay. So the ORC complex is bound to this replication origin site, and it's important to remember that at this point our helical structure within our DNA still retains. We haven't broken any hydrogen bonds yet. So shortly after this, it recruits the CDC6 and the CDT1 constituents, okay, and they're going to act as like a loading platform, if you like, for our MC-M27 dimers, okay. And all I mean by that is our double hexameric helicases. They're they're loaded back to back. While we say double, there's 2 of them loaded back to back in a dimer, okay, and they're going to sit on top of the CDC6 and CDT1 constituents. And now we're still in our G1 phase, and the ORC, along with the CDC6 and the CDT1 and our MC27 dimers, that essentially makes up our inactive pre-replication recognition protein. So now we move to the S phase of the cell cycle, or the synthesis phase. The 2 enzymes, cyclin-dependent kinase and D-dependent kinase, are going to work together to activate and regulate this protein constituent respectively. So D-dependent kinase will directly phosphorylate both the MC2 7 proteins. So upon phosphorylation, those MCM27 proteins will recruit their CDC45 and GINS constituents respectively, and that phosphate coupling along with that protein recruitment causes that conformational change. Okay, it allows them to become active motor proteins. Okay, they disassemble from that protein complex. They offload, if you like, from the CDT1, CD6 constituents, and they move in a bidirectional manner. They both go in opposite directions and break those hydrogen bonds in the double helix. Okay, and this leaves the ORC and the CDC6 and CDT1 constituents exposed. So that's where cyclin-dependent kinase comes in. It phosphorylates the remaining constituents, so it phosphorylates that CDC6 and CDT1 loading platform, preventing the relicensing of another MCM27 dimer. So it prevents the reloading of another 2 helicases. Okay, it adds some level of regulation. Okay, we've already had 2 offloaded and now breaking a double helix. We don't need another 2. So shortly after phosphorylation by cyclin-dependent kinase, those constituents disassembly, if you like, and then they're recycled in the metabolism, which we won't get into too much. So now we've got our helicases, we've got our CMG, our active helicases running across the double strand. But what's happening now they're running across the double strand and they're breaking that strand is we've got supercoiling and we've got torsonal strain down or upstream from where those helicases are working. So eventually they'll come to a standstill because the DNA will just be too tangled to break it apart. So that's where our topoisomerase type 1 enzyme comes in. It relieves this torsonal strain, and how does it do that? So in its active site, it's got the amino acid tyrosine, okay, and tyrosine in its R group has that alcohol ring, that phenyl ring. That hydroxyl group on the very top of the phenyl ring is going to act as a nucleophile, and it's going to perform a transesterification reaction. Okay, so it's going to form a temporary 3' phosphotyrosal intermediate. So with that tyrosine, that hydroxyl group on the tyrosine is taking that phosphate, okay, from that 3', that 3' phosphate, because it's a phosphodiester bond. Then the downstream nucleotide, that 5' carbon, the hydroxyl group of that 5' carbon on the negative oxygen is going to want to take that bond back, and it kind of causes it to rotate, okay, and then it takes that ester bond back, okay, in an ATP independent manner. So this enzyme is ATP independent. That bond is taken back, okay, and we get a transesterification reaction, and in the process of rotating it relieves that torsonal strain, and it allows helicase to continue to move up and continue to break our parent strands so they can be used as template strands to form the 2 daughter strands. Now, we also can't forget the role the single-stranded binding protein plays. So as we've opened our strand of DNA, the single strand of binding protein assembles on that phosphate sugar backbone of both parent strands to prevent enzymes like topoisomerase from getting confused and breaking that backbone of the strands that we want to use, of the open strands that we want to use as template strands. Okay, so that's when the single-stranded binding protein comes in. Okay, whereas topoisomerase type 1, it's relieving supercoiling of the helical, that retained helix structure down or upstream from where helicase and DNA polymerase are working, because they're forming these replication bubbles, if you like. So that's the single-stranded binding protein is protecting those template strands. So now we're going to speak about the next phase of DNA replication, which is elongation. So the transition from initiation to elongation is essentially marked by the introduction of the primase enzyme and the use of RNA primers to actually start building our daughter strands from the template strand. So primase is a really important enzyme because it synthesizes those RNA primers de novo. So what does that mean? It synthesizes them from scratch. And what RNA primers essentially are short ribonucleotide mini polymers, if you like. So they're usually about 10 ribonucleotides in length, and they're transcribed they're synthesized from the primase enzyme. Okay, and the primase enzyme it's the same principle as DNA polymerase. The primase enzyme is using the two parent strands, the two template strands, to essentially assemble a short strand of RNA where the daughter strand will be built from. Okay, DNA polymerase requires those hydroxyl groups, remember, because it's synthesized in a 5 to 3 prime direction. We require those that nucleophilic kind of exposure, those hydroxyl groups to act as nucleophiles to assemble our polymer. Okay, and primase is a really important enzyme because it can do this de novo. DNA polymerase cannot build DNA de novo because of that need for the hydroxyl group, because of the chemistry behind the formation of the DNA polymer. So just to reiterate, the primase enzyme reads a template in a 3 to 5 prime direction, but it synthesizes RNA primers in a 5 to 3 prime direction. And I haven't really spoke about the structure of RNA yet, but I will speak about it more in depth when we look at protein synthesis. So now I just thought we'd kind of get into the why. So why can this primase enzyme synthesize short RNA primers de novo, but our DNA polymerase requires RNA or the use of RNA primers to build its DNA strand, so it can't synthesize a DNA polymer de novo, and it all comes down to the structure of the sugar. So as we know, to take a few steps back, DNA is a polymer. Okay, it's made of many monomeric subunits. Okay, repeating monomeric subunits. Okay, and they're called monomers. In this case, we've got our deoxynucleotides. Okay, so a deoxynucleotide monomer consists of a nitrogenous base, the deoxyribose sugar, and that phosphate group that's linked to the sugar via a phosphoester bond. Now, RNA is also a polymer, which we'll talk about more when we go over protein synthesis, okay, and transcription and translation. If you were to, so RNA being a polynucleotide, another polymer, if you take one of those subunits out, you get a nitrogenous base. You also get a pentose sugar. Okay, so they're both 5 carbon sugars, and you get a phosphate. But the only difference is that pentose sugar is a ribose sugar, so it's got an additional oxygen. So on its second carbon, it's got that hydroxyl group. And I apologise because my head's actually covering the deoxyribose sugar. But on this where it says ribose in the arrow next to my head, that's an example of a ribose sugar, and it's pointing to that oxygen. If you were to look at a deoxyribose sugar, that oxygen's absent and there's just a hydrogen there. So if you're looking at a mononucleotide, always look at the sugar, and that will allow you to infer whether it's RNA or DNA. If you look at that second carbon hydroxyl group. And just to take another few steps back, the picture above I'm showing is a example of a ribonucleotide monomer. So we've got our uracil nitrogenous base, okay, and uracil is replaced with thymine. Uracil replaced with thymine in RNA. Okay, and thymine is present in DNA. Now, remember when we looked at the structures of the nitrogenous bases individually and we compared them in the first video, we've inferred that thymine and uracil are very similar in terms of their molecular structure. The only difference is uracil lacks that methyl group on its 5th carbon, and you can see that methyl group is missing. So the methylation of uracil gives you thymine. I think it's thymidylate synthase using folate as a cofactor. But anyway, that's not relevant. What the main thing I'm trying to communicate here is the main difference is in that sugar, that second hydroxyl group on the carbon. Okay, and that's really important. That small difference in functional groups allows for the primase enzyme to synthesize a short strand of RNA or an RNA primer de novo, and it's to do with orientation. Okay, so in order to we know the chemistry behind polymerization of nucleotides, whether it's RNA or DNA, we need that 3' hydroxyl group to act as a nucleophile to an incoming deoxynucleotide triphosphate or a ribose nucleotide triphosphate in the case of the primase enzyme. So that 2 hydroxyl group, that 2' hydroxyl group doesn't participate in phosphoester bond chemistry. That's not what I'm saying. But what I'm saying is for that oxygen allows for the correct hydrogen bond to form between the primase, okay, and its substrate. It allows for the correct rotational positioning of that nucleotide triphosphate, of the ribonucleotide triphosphate, or the correct orientation for that 3' hydroxyl group to be exposed. Okay, so it allows for that exposure to allow for that initial polymerization to occur, for that hydroxyl group to accept that alpha phosphate on the incoming ribo nucleotide triphosphate. Okay, if you remember, those phosphoanhydride bonds between the beta and the gamma phosphates are cleaved, which allows for that work to happen. And again, primase has magnesium in its active site, so magnesium stabilizes that enzyme substrate complex, okay, and it reduces that electrostatic repulsion between the negatively charged hydroxyl group that we've exposed, okay, that 3' hydroxyl group in the enzyme substrate complex, and the incoming ribose nucleotide triphosphate. And those phosphate groups, that gamma and beta phosphate, which are cleaved, and we get pyrophosphate. And essentially that stepwise phosphorylation process and the polymerization process we discussed in the video before is pretty much identical, but I'm going to go over it again in the next few slides just so we can build a picture. We're just taking a few steps back. So we're looking at how RNA primase mechanistically synthesizes an RNA primer strand de novo. Okay. So just to kind of recap on this stepwise phosphorylation process, if this is completely alien to you, then I recommend going back to the previous video where we discuss nucleosides to nucleic acid ready nucleotides, if you like. But I'll just give a quick recap, just because we didn't discuss it for RNA, we only discussed it for DNA. So just go all the way back. Okay, so just to recap, so stepwise phosphorylation is a process in which a nucleoside, so a nitrogenous base bonded to its sugar, whether it's deoxyribose or ribose sugar, okay, becomes a RNA or DNA ready nucleotide. So in this case, we're going to talk about ribonucleotides. So we start off with our nitrogenous base and then our deoxy and our ribose sugar. So you can see each of the sugars on the top, whether it's adenosine, guanosine, uridine or cytidine, all have those hydroxyl groups on the second carbon. Always look out for that second carbon, and that will indicate whether it's a ribose sugar or a deoxyribose sugar. Again, just to reiterate, both deoxyribose and ribose are pentose sugars, so they're 5 carbon sugars, but the only difference is their functional group on that second carbon. So anyway, in order for a nucleoside to become an RNA-ready nucleotide, we need to do a 3-step phosphorylation process. And this 3-step phosphorylation process requires ATP as a phosphate donor each time. It requires kinase enzymes, and it requires that magnesium in its active site to stabilize that charge repulsion, okay, to act as a charge shield. So I'm going to use uridine as an example because we already discussed adenosine, guanosine, and cytidine. So we start off with our uridine nucleoside, which is just uracil bonded to our ribose sugar via a glycosidic bond. Okay, and then uridine kinase is going to transfer a phosphate group. ATP, adenosine triphosphate, is acting as our phosphate donor. It's going to catalyze that transfer of phosphate groups to give us uridine monophosphate, and we get adenosine diphosphate as a product. Okay, now the adenosine monophosphate and the guanosine monophosphate and the adenosine monophosphate and the cytidine monophosphate, just to reiterate, it's exactly the same reaction, just different enzyme name, okay, but we get the same kind of monophosphate and a different nitrogenous base. So for adenosine, it's adenosine kinase, for cytidine, it's cytidine kinase, and for guanosine, it's guanosine kinase. So we've now got our uridine monophosphate. So now with the help of uridylate kinase, and again with ATP as a phosphate donor, and that magnesium in its active site acts as a charge shield between the repulsion experience between that alpha phosphate on our monophosphate, okay, and the incoming ATP, the beta and gamma phosphates on the ATP is going to act as a charge shield. Okay, we get uridine diphosphate, so we've now got 2 phosphate groups. So remember, we formed a phosphoanhydride bond, so we haven't formed a phosphoester bond. We formed a phosphoester bond when we got uridine monophosphate, that ester bond between that ribose sugar and our phosphate group. Now we formed a phosphoanhydride bond between the alpha and the beta phosphates, and we've got ADP, adenosine diphosphate, as a byproduct. And again, it's the same principle for the other monophosphates: adenosine monophosphate, cytidine monophosphate. ATP is always that phosphate donor. Magnesium is always a charged shield, and the kinase enzymes, whether they're cytidylate or adenylate or guanylate, will always catalyze that transfer of phosphates. So now we've got our uridine diphosphate. So it's got our two phosphate groups. We've got our alpha and beta phosphate. The alpha phosphate's linked to the ribose sugar via a phosphoester bond, and then the alpha phosphate's linked to the beta phosphate via a phosphoanhydride bond. Now we've got our universal phosphorylator. And remember, I use the term universal loosely, and this is the same if you remember the stepwise phosphorylation process for the deoxy ribonucleotides. Okay, you'll remember it's that same universal phosphorylator nucleoside diphosphate kinase, and that will phosphorylate. That will transfer the phosphate group from ATP. So ATP is still our phosphate donor. Okay, the enzyme uses that magnesium as a charge shield because there's repulsion between the beta and gamma phosphates in the ATP and our phosphates in our ribonucleotide diphosphates, if you like. Okay, so that magnesium's acting as a charge shield, and we've catalyzed the transfer of another phosphate group. So ATP is our phosphate donor. It's therefore lost the phosphate group, so it becomes adenosine diphosphate as our byproduct, and our main product we get is uridine triphosphate. Okay, so that's the uridine's now got 3 phosphates, and again, I'm only speaking about uridine here because we didn't speak about uracil in the previous video, so I'm just letting everyone know it's exactly the same example, and it's the same, for example, with guanosine diphosphate. With the help of nucleoside diphosphate kinase and ATP as a phosphate donor, we get guanosine triphosphate. Okay, so now we've got our uridine triphosphate. It's ready to become a RNA ribonucleotide for our primer. Okay, so our primase enzyme can now use that it can now use that as our primer, if you like. So now we've got our ribonucleotide triphosphates. They're ready to kind of polymerize now. They've got that phosphoanhydride bond is going to allow for that work to happen. Now, what essentially primase is working the same way as DNA polymerase. So DNA polymerase reads the template parent strands in a 3 to 5 prime direction, and it assembles the nucleotide polymer in a 5 to 3 prime direction. So we want that hydroxyl group exposed to act as a nucleophile to accept that alpha phosphate, okay, from the incoming ribonucleotide triphosphates. Okay, and the reason we did that stepwise phosphorylation process is because we need that phosphoanhydride bond to give us that free energy to allow for that bond formation to occur. So it's reading the two primase enzymes. Okay, they're also moving bidirectionally across the 2 parent template strands, just to link this into DNA replication. Helicase has split our double helix. Okay, and now the primase enzymes are moving in opposite directions across the template parent strands. They're reading in that 3 to 5 prime direction. Okay, and they're assembling short RNA primers in what's called a complementary fashion, the same way DNA polymerase is assembling our daughter DNA polymers in a complementary fashion. Primase is doing the same thing. So let's say our parent template strand, we're reading it 3 to 5, just to reiterate. Okay, it's adenine, right? The parent strand is expressing adenine. Then the corresponding ribonucleotide on that primer is going to be uracil. Okay, it's not going to be thymine, it's going to be uracil, because uracil replaces thymine. Now, if it's reading thymine on the parent template strand, there's going to be adenine on the corresponding RNA primer. If there's cytosine on that parent template strand, then guanine is going to be that corresponding ribonucleotide on the RNA primer. Okay, and then similarly, if guanine is on that parent template strand, then cytosine will be on that RNA primer. So I'm showing this diagram again because I think it's a really good indication, and we can go back to it and it all becomes kind of clear now. So we've got our kind of replication bubble, okay, and we've got helicases moving in the direction of the replication fork, and depending what way the replication fork is going will dictate what strand is what. So we've got our leading strand and we've got our lagging strand. Okay, but there are differences in how the daughter cells, the daughter strands are formed by both parent templates. So on the leading strand, it's a very simple process, okay, because it's going the same direction as the replication fork, okay, and the enzyme's reading, they're both still reading, the DNA polymerases, the primases, they're all reading in a 3 to 5 prime direction. But because of the anti-parallel nature of DNA, if you're looking at it as you're splitting the strands, they're going in opposite directions, but they're actually reading in the same direction, and they're assembling their respective daughter strands in the same 5 to 3 prime fashion as well. And this is all to do with that's understanding the anti-parallel nature of DNA. And essentially, so for the leading strand, it's quite simple. Primase will place a short RNA primer, okay, and DNA polymerase is able to assemble a long daughter strand, okay, using that RNA primer, using that exposed 3' hydroxyl group from the RNA primer to then when it adds that incoming when DNA polymerases adds its incoming deoxynucleotide triphosphate, okay, that deoxynucleotide triphosphate is going to have its 3' hydroxyl group exposed, ready for another, acting as a nucleophile, ready for another incoming deoxynucleotide triphosphate. So it can form a long daughter strand without any problems, but the lagging strand is a bit different because it's going away from the replication fork. So it requires RNA primers strategically placed across the daughter strand, if you like, to allow, or where the daughter strand is going to be formed, to allow for short DNA fragments to form in between those RNA primers. So they're called Okazaki fragments. Okay, so those Okazaki fragments essentially are formed across the lagging strand. Okay, so you get what's called a DNA RNA hybrid, where there's RNA primer, Okazaki fragment, RNA primer, Okazaki fragment. Okay, and they're just continuous strands of DNA that are formed of the daughter strand that are formed in between RNA primers, and they use primers to kind of develop on. Okay, so DNA polymerase is synthesizing these Okazaki fragments. So now we've got our RNA primers strategically placed. On the leading strand, we've just got one primer, and then we've got a long daughter strand of DNA being formed by DNA polymerase. On our lagging strand, we've got RNA primers strategically placed across where the daughter strand is going to be formed, adjacent to the template strand, okay? And then those Okazaki fragments of DNA are forming in between those RNA primers. So we're building a daughter strand using both parent strands. So both the 3 to 5 and the 5 to 3 strands are being used as parent strands respectively. And as DNA polymerases is forming these daughter strands, not only is it reading a template strand in a 3 to 5 direction and then forming a DNA polymer in a 5 to 3 direction, it's also proofreading along the way. So it's a very clever enzyme. So it's doing multiple things at once. Okay, and the concept of proofreading ensures genomic integrity essentially, so correcting misincorporated nucleotides, because these enzymes do make mistakes, and a lot of this we'll talk about this in the videos to come when we talk about DNA mutation, substitution, insertion, and all that deletion. Okay, a lot of these mistakes, proofreading prevents a lot of these mistakes from happening. So enzymes respond to geometry and charge. Okay, so they can recognize distortions in the helix based on this charge, a change in charge and geometry, okay, from what it's supposed to be based on what the template strand is reading. And so, for example, if polymerase it recognizes that it has a it recognizes it has a mistake in its pairing, then it will activate its exonuclease domain. So its polymerase domain is responsible for reading the template strand, and it's responsible for putting together or assembling the daughter strand of DNA. The exonuclease domain is responsible for removing nucleotides, removing misincorporated nucleotides. And how it does this is via hydrolysis. So it uses the magnesium in its active site to essentially further polarize a water molecule, and then that water molecule is used to break that phosphodiester bond of the incorrect nucleotide, okay, and then it will replace it with the correct nucleotide. It will proofread once again and then move across, and you can see the error rate in the table without proofreading and the error rate with proofreading. There's a significant difference, so it emphasizes the importance of proofreading via DNA polymerase. So this is the structure of DNA polymerase. The main thing we're interested in is the you've got the fingers which bind to the incoming deoxynucleotide triphosphates, okay, and then you've got the polymerase active site, okay, and that actually synthesizes our strand of DNA, okay, in a 5-to-3 like manner, okay, and you can see that you've got magnesium in that site. That's to reduce that charge repulsion that we experience between the incoming DNTPs, that phosphoanhydride bond that holds the beta and gamma phosphates, and that phosphate or that hydroxyl group that's being exposed, that negatively charged hydroxyl group that will be bound to the blue part, the palm, if you like, and that will assemble together in a daughter strand in a polymer. It's the kind of chemistry behind it. And then you've got the exonuclease domain in purple, okay, and that's got another magnesium in that site to kind of activate this water molecule, if you like. And when it detects a mistake, it will flip, and the exonuclease domain will be active, and it will break that phosphoester bond that we formed on our growing strand to get rid of that nucleotide, and then to replace it, okay, it will flip back to the polymerase active site, and then that polymerase active site will replace it with the correct one. It will proofread again just to make sure, and then it will move on. So now we're going to just quickly touch on RNA primer removal. In terms of, I've put more information than you actually need to know, but I think it's important to just kind of conceptualize what's going on here, because I think there's a lot of conflation in literature. So RNA primal removal is predominantly done by two enzymes. That's RNase H and Fen1. So RNase H removes the majority of ribonucleotides. Okay, so as you know, a primer is made up of, say, 10 or 15 ribonucleotides. Okay, RNase H removes the majority of those primers. Okay, Fen1 will remove the primers where one RNA ribonucleotide meets a DNA nucleotide. Okay, so just to kind of conceptualize this, so let's say you've got on our lagging strand, I've put on the left-hand table down below, I've put DNA, 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 RNA, 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 DNA, 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 to emphasize that kind of hybridization. So we've got the DNA, 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 they're the Okazaki fragments, okay? We've got the RNA, 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 that's our RNA primer. So RNase H will remove the internal ribonucleotides. So it will remove those two RNAs in the middle, okay, and that will give us DNA, 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 RNA, X, X, RNA, DNA, DNA, DNA. The X, not to confuse anybody, I've just had X. Well, X just means there's nothing there now, so we've removed those two RNA primers. So then another type of polymerase enzyme is going to come in, okay, called delta polymerase, and that's going to start assembling using template, using the parent strand as a template. It's going to start assembling DNA DNTPs, okay, in a 5-to-3 prime fashion. It's still reading the template in a 3-to-5 prime fashion, but it's assembling those DNTPs in a 5-to-3 like fashion, because we need that hydroxyl nucleophile on that 3 prime. Okay, and what's happening as it's growing that chain, those RNAs that are linked directly to the Okazaki fragments, they become displaced, and it causes what's called a 5 prime flap. So they get displaced, okay, and then Fen1 will come along, and it will directly remove the 5 prime flap, and then the growing strand of DNA will form, okay. But you've still got a gap between where the newly formed DNA by delta polymerase is and the Okazaki fragments that are already existing at this point. There's still a gap because that phosphate sugar backbone isn't linked yet. Okay, so that requires another enzyme to kind of bridge that gap. And before we move on to that enzyme, I'll just kind of summarize this table. In terms of this process, it's not really overly emphasized in literature. I guess the main takeaways are just know that RNase H and Fen1 will remove those primers. I'm just giving you a bit of background on how they actually do it. This is just another diagram to kind of emphasize the process, and it's included DNA ligase, which I'll talk about in the next slide. So you've got in purple in the first one, you've got your RNA primer, okay, and RNase H you can see is removing those internal ribonucleotides, okay, and it's leaving the ones that are directly linked to the Okazaki fragments, okay. And then what's happening is delta polymerase or DNA polymerase is coming in, and it's refilling in that gap. It's still reading in a 3-to-5 prime direction, but it's assembling in a 5-to-3 prime direction, those incoming DNTPs, those deoxynucleotide triphosphates. and this causes those and I like to think of it this way: the delta polymerase doesn't care that there's a ribonucleotide linked to the Okazaki fragment. It's just going to put the respective deoxy nucleotide triphosphate there anyway, and that causes that ribonucleotide that's linked to the Okazaki fragment, that downstream deoxynucleotide that's linked, that starts the Okazaki fragment, it's linked to that, it's going to get displaced and it's going to form a 5' flap. Okay, and then Fen1 will come and remove that flap, but then we have a gap between what was the Okazaki fragment and what now is this new form of DNA where that primer used to be, and DNA ligase is going to come, and that gap is because that phosphate backbone isn't linked. DNA ligase is going to come and it's going to fill that gap. It's going to form that phosphoester bond or phosphodiester bond, okay, and it's going to join that strand, so you'll have your daughter strand of DNA. And this is predominantly on the lagging strand, because as you know, there's multiple primers used on the lagging strand, and the Okazaki fragments formed in between those primers. So DNA ligase is the enzyme we just spoke about in the previous slide. We'll talk about it now. It's an ATP a dependent enzyme, so it's dependent on ATP, and it's the enzyme responsible for forming that phosphodiester bond. Okay, once Fen1 and RNAs have removed those primers, and delta polymerase has replaced it with a with a short strand of DNA instead. And how it does this is through ATP essentially. So the ATP in its active site, once hydrolyzed, will form what's called an AMP or adenosine monophosphate intermediate. So it forms this 5' phospho AMP intermediate. Okay, so that means a downstream 3' OH on that on the Okazaki fragment. Okay, so just remember we've removed it. The AMP, the 5' phospho AMP intermediate is formed with the nucleotide that used to be where that ribonucleotide would be joining with the Okazaki fragment. Okay, so the downstream 3' hydroxyl group and that Okazaki fragment becomes activated, and that phosphate, that 5' phosphate, okay, that AMP is displaced. So it will then form a phosphodiester bond. Okay, and magnesium juice stabilize the repulsion between the 5' phospho AMP intermediate and that hydroxyl nucleotide on the that start in that kind of what used to be the Okazaki fragment, and that essentially allows that phosphate sugar backbone to form. Okay, and that means they're linked. That means they're now linked. That new strand of DNA that delta polymerase has synthesized and that Okazaki fragment are now linked, and we've got our daughter strand of DNA now on the lagging strand. So just to kind of do a quick recap, I've got a lot of information here just in case anyone's confused. All this information you need to know is here, and you can pause it and take it down. But just to recap, we've got our initiation, which starts in the G1 phase of the cell cycle. So this is the assembly of our inactive pre-replication protein complex. Okay, so that starts off with the ORC complex, the origin recognition complex, and that recognizes the origin of replication sites on the DNA double helix. They're those adenine thymine rich areas that are less thermodynamically stable because of the hydrogen bonding. Okay, and this ORC binding to these areas allows for the recruitment of the CDC6 and CDT1 constituents respectively, and they act as a loading platform for our MCM27 dimer complex, which is just our 2 hexameric helicases in active form loaded back to back. Okay, as we move into the S phase of our cell cycle, okay, cyclin-dependent kinase and D-dependent kinase are going to activate and regulate this protein complex respectively. So D-dependent kinase will phosphorylate both the double-loaded hexamers, so both the helicase enzymes. This phosphate coupling results in a conformational change. It allows them to recruit their GINS and CDC4/5 complexes respectively. Okay, and they then both become active motor proteins, and they'll offload from the CDC6 and CDT1 platforms. They'll go in a bidirectional manner, and they'll run across, breaking that double helix strand. Cyclin-dependent kinase will quickly come along and phosphorylate the remaining ORC, CDC6 and CDT1 constituents to prevent the relicensing of another MCM27 dimer. Okay, and as helicase, as both the helicases, the CMG helicases, are running down the double helix, they're breaking those bonds, but downstream or upstream, where you've got the double helix still intact, it's resulting in torsonal strain, so eventually, and supercoiling, so eventually helicase is going to come to a standstill, but that's where topoisomerase type 1 comes in. Okay, it's relieving that torsonal strain downstream. Okay, and its tyrosine in its active site is the key to how it does this. Okay, so it does this through a transesterification reaction, so it's stealing that ester bond temporarily. Okay, so it forms a 3' phosphotyrosal intermediate, so that alcohol ring, that phenyl ring on its R group, is acting as a nucleophile, allowing it to steal that phosphate, okay, forming a 3' phosphotyrosal intermediate. So then a downstream nucleotide, that 5' hydroxyl group in a downstream nucleotide, has just lost its ester bond. It's going to want it back, and this causes it kind of rotation. This causes like a rotation as the bond is reformed, and then it relieves that tangling or that supercoiling, and it allows helicase to continue to run across. As helicase is breaking those hydrogen bonds, okay, we've now got our 2 template strands, okay, and in order to prevent them from reannealing and coming back together, so that DNA polymerase can work on them and primase can work on those template strands, the single-stranded binding proteins will bind to the phosphate backbone, and not only do they prevent reannealing, they also prevent nuclease enzymes like topoisomerase 1 from degrading those phosphate backbones, okay? And that's essentially our initiation phase. Then we move into our elongation phase, where we're starting to now synthesize our daughter strands using the template strands. So we need RNA primers to do this, and that's where our primase enzyme comes in. It can synthesize RNA primers de novo because of that hydroxyl group on the second carbon of the ribose sugar that the deoxyribonucleotides don't have. They just have the hydrogen. It allows for the correct enzyme substrate rotation, if you like, to expose that 3' hydroxyl group to then build an RNA primer. Okay, and just to be clear, remember RNA primase is reading the template strand in a 3 to 5 direction, but it's forming RNA primers in a 5 to 3 direction, because we know the chemistry of nucleic acid polymerization relies on that hydroxyl group to act as a nucleophile. So our incoming either ribonucleotide triphosphates or deoxyribonucleotide triphosphates. But in the case of the RNA primase, it's incoming ribonucleotide triphosphates. So it's synthesizing short RNA primers complementary to the template strands, okay, and this allows for DNA polymerase to then build off of that. In the leading strand, it will build a continuous DNA polymer daughter strand using that template strand, continuing in a 5 to 3' fashion, building that strand but reading a template strand in a 3 to 5 like fashion. In the lagging strand, RNA primers are placed strategically across, corresponding with a template strand, in order to allow DNA polymerase to form these Okazaki fragments between these RNA primers. So it's doing it in a fragmented manner, if you like. So don't forget as well, DNA polymerase contains its exonuclease domain, which allows it to kind of proofread along the way. So on both leading and lagging strand, proofreading is happening as it's creating and reading, as it's reading the template strands in a 3 to 5 direction, creating the daughter strands in a 5 to 3 direction. It's also proofreading the daughter strand, okay? If it senses any differences in geometry and charge, density, or whatever, then it's going to activate its exonuclease domain, okay? That phosphoester bond and the forming on the new daughter strand is going to be hydrolyzed, and that wrong nucleotide is going to be removed, and it's going to be replaced with the right one. Okay, so now we've formed on our lagging strand, we've got Okazaki fragments and RNA primers. We've got an RNA DNA hybrid. Okay, so this is where our RNase H enzymes and our FEN1 comes in. So as you know, RNase H removes those internal ribonucleotides. Okay, and then delta polymerase comes in. Okay, it builds a DNA polymer, same principle, in a 5 to 3 direction, or short DNA kind of polymers where those internal primers used to be, reading the template in a 3 to 5 direction, but synthesizing them in a 5 to 3 direction, because we need that nucleophile exposed. Okay, and this causes those ribonucleotides on the terminals of the primers that are linked to the deoxynucleotides that start our Okazaki fragments. Okay, they become displaced, and then FEN1 forms a 5' flap. FEN1 comes in and it removes that 5' flap, and then ligase comes in, used with the help of ATP, forming that 5' phosphate AMP intermediate, which allows for that downstream 3' hydroxyl nucleotide on the Okazaki fragment to form that phosphoester bond with the backbone. So you get ligation of that backbone, and we've got our 2 daughter strands of DNA. So that is initiation and elongation summarized up. I know it's a lot to remember, and it's more so remembering the proteins and the enzymes and stuff, but that's essentially that process summed up. Now we've got the termination process, which we'll discuss in the next slides. So now we've got the final stage of termination. So you get what's called fork convergence. So the the two helicases, the two helices structures become intertwined with each other, and that's where topoisomerase type 2A comes in. I'm not going to really touch on this too much because we already went over topoisomerase type 2A when we looked at topoisomerase type 1. But as you know, it's a homodimer. It's got two domains, those nuclease domains. It works, it requires ATP to cause that conformational change, but it's still a transesterification reaction. We're not forming any phosphointermediates, we're forming those 5' phosphotyrosal intermediates. It's still got that tyrosine in its active site, it's still got that phenyl ring, that alcohol. It's the same principle, and it just decatenates the strands. It works the same way as topoisomerase type 1, only it's got two domains for each helix, if you like, and it's bonding to one backbone on one helix and another on another helix, and that rotation will allow for decatenation. But there's a problem, okay, with the lagging strand. So that's that kind of topoisomerase 2A's role. But now we're going to just talk about the lagging strand, okay, and we're going to talk about telomeres and telomerase enzymes. So once on the lagging strand, once that final 5' flap is removed, there's no 3' hydroxyl group. There's nothing, okay. The telomeres take the fall, okay. They take the fall. There's a non-coding DNA, and they take the fall at the end of the chromosomes. And on the lagging strand, there's nothing to build off of. And this is due to the removal of that last primer. And as you know, it can a polymer, a nucleotide, can only be formed in a 5' to 3' direction. You need that hydroxyl group to act as a nucleophile. Otherwise, you cannot form your polymer, or you cannot even form your primer. But there's nothing there to go by. Sorry, I meant if you can form your primer, but if you remove that primer, it leaves you with the same problem as you started, right? We cannot form DNA de novo without the use of RNA primers, okay? Because of the way in which, because of that hydrogen on that deoxyribose sugar, the orientation between the enzyme and the substrate does not expose that hydroxyl group in the way we need it to be exposed. So this is where telomeres and this is where telomerase enzyme comes in. Okay, so the telomerase enzyme itself, so in the diagram what I've shown is what's called a 3' overhang. Okay, so that parent strand has all the right DNA sequences, okay, but that daughter strand is missing, it's missing a significant amount of sequences. But telomerase actually has its own RNA transcript built in. Okay, it's got an AAUCCC transcript, and this corresponds with the telomeres sequences. So telomeres are non-coding DNA, and they take the fall for gene loss, okay, to prevent actual genes from getting lost during replication. And telomeres have a TTAGGG, okay, like sequence. So complementary to that would be an AAUCCC. And all telomerase does is it binds to the 3' overhang, okay, and it uses that RNA AAUCCC, it's really clever, as its own sort of like template for DNA polymerase to then build to then refill the missing information. Now there's a lot of kind of conflation in literature, and I won't go over it too much because there's a lot that you probably don't need to know. But that delta polymerase enzyme, that same enzyme that was filling in the removal of once the primers removed, that same enzyme that was replacing that removed primers with actual DNA fragments, will do exactly the same, only telomerase is acting as a transcript, and this is called reverse transcription. So we're making DNA using RNA as a template, and you can see the AAUCC corresponds with the TTAGGG. So we've got thymine, we've got adenine. Well, we've got adenine, we've got the uracil on telomerase RNA transcript. Well, we've got the guanine, we've got the free cytosines. So, and it's important to remember that the in telomerase, the enzyme, it's a clever enzyme, right? The RNA transcript isn't used up. That's not what I'm saying. It's not placed on that daughter strand. It's used as a template for then delta polymerase to come in there and then build on from that daughter strand to prevent drastic loss of telomeres, okay? And it's very effective in doing so. So this is just to essentially explain just to essentially give you another diagram, just to kind of recap what's actually going on here. So you can see we've got our replication fork. The fork's moving in that direction. So we've got the lagging strand is moving away from the replication fork. So it's moving in the opposite direction. We've got our leading strand moving towards the replication fork. Okay, we've got our DNA polymerase enzyme. It's going across the parent strand, reading it in a 3 to 5 prime direction, but it's assembling its daughter strand in a 5 to 3 prime direction. Remember, because we need that 3 prime hydroxyl group to act as our nucleophile. It's using the primer. You can see the primer's in green on the leading strand. It's using that initial primer that was created by primase de novo to then build its own strand of DNA. Okay, it uses it as a starting point. Okay, it needs that initial 3 prime hydroxyl exposure in order for it to polymerize the incoming deoxynucleotide triphosphates. Okay, and our helicase enzyme is in orange. That's breaking the strand. You can see topoisomerase type 1, that would be, is in blue. That's working ahead of helicase, and that's alleviating the torsional strain that's caused by helicase running across that helix structure. Our lagging strand is moving away from the replication fork. It's important to remember, though, even though it's moving away from the replication fork, DNA polymerase is still reading the strand in a 3 to 5 prime direction, and it's still synthesizing the strand of DNA in a 5 to 3 prime direction. Primase is doing the same thing with its primers as well. Okay, but it just does them in fragments, and then the Okazaki fragments are formed between those primers. They'd then be subsequently removed by RNase H and FEN1. So RNase H removes those internal ribonucleotides, but not the ones that are directly linked to the deoxynucleotides, we'll call them, that will start that Okazaki fragment. The FEN1 is removing the 5' flap, okay, that's caused by displacement of nucleotides or deoxynucleotides via delta polymerase. So delta polymerase is coming in there after RNase H has removed those, and it's just building a short strand of DNA. It doesn't care they're still ribonucleotides linked to those Okazaki fragments. It doesn't care. It's just going to build them anyway. They're displaced, and FEN1 removes them. Okay, and then you get our ligase comes in with the help of ATP and those phospho-AMP intermediates. Will then religate that phosphate backbone, and you get your 2 daughter strands. And as you know, in termination, you get the 5' overhang on the lagging strand, and to prevent gene loss, telomerase comes along with its own transcript of RNA to use as a template for reverse transcription for delta polymerase to then fill in those gaps on the daughter strand. And that's essentially DNA replication. I hope you found this video helpful, and I'm aware it's very long. We didn't just go over DNA replication, we went over the double helix as well and the concept of hydrogen bonding. But in the next video, it's likely we're going to be looking at DNA mutations, and then we're also going to be looking at protein synthesis, transcription, translation, and then we're looking at the structure of amino acids themselves. So yeah, stay tuned, guys, and good luck with your studies.